This program is brought to you by Stanford University. Please visit us at stanford.edu.
So I want to talk a little bit about the science of stem cell research first, because that's where the government intervention didn't go. And so it's important at least we understand, hey Ed, the science of this, and then move on to what the politics have become. The beginnings of stem cell research I would put at the bombing of civilian populations in Hiroshima and Nagasaki. In retrospect, those people who died at the lowest dose of a radiation that kills died about 15 days after radiation. And when experiments were done on mice and dogs to replicate it, they died because their blood-forming system was destroyed. At higher doses, you have shorter times to death and other body systems are affected. But this really intrigued the community of hematologists and experimental biologists to try to figure out why was it that the bone marrow, the blood-forming organ, was destroyed. I think it's safe to say that one of the key experiments was done in 1961 in Toronto, where indirectly James Till and Ernst McCulloch showed there had to exist in the bone marrow a cell type which had an unusual property. The cell, when it divided to give rise to two daughter cells, gave rise to another cell just like itself, a stem cell. Whereas the other cell, the other daughter cell, started to form blood, and eventually, as we learned, a single stem cell, a single mouse blood-forming stem cell, can end up making over 30,000 stem cells in the body when it regenerates the irradiated host, and make 1 billion blood cells every day. It does it for the life of the animal, and then you can transfer another stem cell to another animal for its life and its life and its life. So it's really a remarkable cell. That opened up the area to try to find that cell rather than know indirectly that it existed. That's where we came in, and I won't go into, I think you'll be grateful, I won't go into the details of how we did it, just to say that we found a way to isolate these cells, and it is, in general, the way you can isolate stem cells from brain and from cancers and other tissues. They are rare cells, about 1 in 20,000 cells in the bone marrow. We found out in mice that we could isolate these cells pure, and that we could save other mice, even genetically unrelated mice, from that radiation damage. And the way that it worked is those stem cells regenerated the bone marrow, the blood-forming organ, replaced themselves, and now grew up in a different host. So if John were another mouse and I was this mouse, my stem cells growing up in his body would make my blood-forming system and my immune system in him. One of the great properties that we eventually showed was that those stem cells growing up in his body would tolerate him. That is, they would not make an immune reaction against him, because as they grew up from the most primitive cell, they accepted him as self. But because they came from me genetically, they would express all of my genes, and they would tolerate me as self. So immediately when we did this experiment, I hate to say that I actually did this experiment the first time in high school in Great Falls, Montana, where I irradiated mouse seats. Old habits die hard. I just keep doing the same experiment again. That I found that you could transplant skin from the donor into the host without any further treatment. And that meant that skin transplants could be accepted. So that was 1957. I went home and told my mom at lunch, I said, you know, in a matter of just 3 or 4 years, people are going to be transplanting bone marrow and hearts and skin and lungs. Well, it's just barely started by now. That was a big lesson, how long it takes to actually try to translate a discovery into medicine. Now, hopefully things are going to go a lot faster now, but I want you to know that I was sure of it, and my mom is still waiting for it to happen. She'd actually like some bone-forming stem cells right about now. So when we were able to isolate those cells and show that transplantation, I was working with a medical student at Stanford, Judy Shizuru, and she was really interested in the problem of diabetes. Hugh McDevitt in the audience. Where are you, Hugh? Hugh McDevitt had shown in the 1970s that diabetes was not a disease of the pancreas and not a disease of insulin-producing cells. It was a disease of the immune system where, because of the genetic predilection of the poor mouse or the poor human that had the disease, instead of censoring the kinds of immune cells that would react against self, they allowed them to escape and proliferate and mature their capacity. So diabetes, juvenile or type 1 diabetes, turned out to be an autoimmune disease, a disease where you destroy your own insulin-producing cells. So Judy Shizuru and I went on to show in the mouse model of the disease that if you simply gave blood-forming stem cells from a parent or sibling mouse, genetically different than the mouse that was just getting diabetes, you could stop that reaction forever and cure the disease. If you waited until the diabetes had proceeded and killed off all the insulin-producing cells, then you needed to give both the blood-forming stem cells from the donor and the insulin-producing cells. Well, that's where we ran into a difficulty that made us think about other cell types, because we didn't have a lot of extra insulin-producing cells. Jeff Bluestone back there, my colleague at UCSF, knows this problem very well. So when it finally became known that there were a class of cells called embryonic stem cells, which I'll come back to extensively, that could make any tissue in the body, we thought maybe there are such things as pancreatic precursors or pancreatic stem cells that could be grown from these very primitive embryonic stem cells. So that led us into the kinds of investigations that started to get us into trouble with the public, that is, working with cells that some people thought were immoral entities to work with. We did it in mice for a long time, so we didn't have a lot of flack about that. Although I can tell you, at many times in my career, I've been criticized by colleagues even here at Stanford for working with mice. I think Dick Lyman will remember some of those times. Anyway, the blood-forming stem cell experiment was a way forward for us to know that at least that tissue could be regenerated from stem cells. Later, we went on to show that you could find brain-forming stem cells, and those brain-forming stem cells could actually, unlike blood-forming stem cells, be propagated outside the body. And that led to, at a company that I co-founded, the transplantation of human brain stem cells into children with a genetic disease which is always fatal. It's fatal because their brains build up a substance they can't degrade, a toxic substance, because they lack one enzyme and they lack one part of a gene that would make that enzyme. Those kids are normal until 1 to 5. This is called Batten's disease. And then they start having cells in the eye and the optic cortex that it leads to die because they build up a fatty protein deposit that pops the cells. And then it happens back in the cerebellum, which governs balance and coordination, so they had what's called cerebellar ataxia. And then they started losing IQ because it was working on the part of the brain that involves learning, the hippocampus. And then they go into a coma, and then they all die. So it's nice to know that those brain stem cells taken from a human and put into the brain of an immunodeficient mouse that had the same genetic mutation could stop the disease in its tracks. The way that they stop it is not terribly important. I'll just say they make the enzyme that's missing, they transport it out into the fluid that bathes the brain, and the cells that are missing the enzyme take it up and break down this potentially toxic product. Now, the reason I'm spending enough time on the detail of this is that we were transplanting human brain cells into the brains of mice. And this has excited a lot of people who have not always been our friends, that said that this was not a good thing to do. All sorts of images of metamorphosis come out. The idea that there might be a human brain stuck in the brain of a mouse. I'll just say that we accomplished maybe 1% of the neurons and the supporting cells coming from the mouse, but it was and still is a very serious allegation. Senator Brownback, and I'll come back to him in a minute, Senator Brownback in Kansas introduced legislation to ban this research, criminalize this research. $1 million fine and 10 years in jail if it had passed, and we certainly do it at Stanford. We have used these brain stem cells to study not only these neurodegenerative diseases, but it actually can reverse most instances in a mouse of spinal cord injury, because the spinal cord is crushed in an injury, as in Christopher Reeve, and the cells whose cell bodies are right in the part that's crushed die. The cells that are up in the brain don't die. The cells that are below it that'll send pain signals up don't die. But because the cells die in that little region, they don't have the insulator that allows the quick electrical transmission of the impulses of pain or motor control. When we put human brain stem cells just above and below that lesion, they remyelinate it, it's what's called. And that leads to the animals being able to walk. So the challenge, when Senator Brownback asked me about this in a committee meeting at the Senate, is which of these neurological diseases, which of these tragedies do you think we shouldn't work on as hard as we can? And he didn't respond exactly to me in the same way that I presented it to him. But it shows that there is a disconnect between those of us who, as MDs, pledged to work on diseases and try to do the utmost that we could, and those people who have different points of view. Of course, everybody's allowed to have their own point of view. As my best friend used to say, everybody's entitled to his own stupid opinion. And a year ago, President Bush, in the State of the Union address, called this kind of transplantation of human brain cells into a mouse's brain as the most egregious kind of experiment that the scientists could do. So it's pretty clear where those lines are. Now, the research that we were doing on normal blood cell development, normal brain cell development, normal lung, breast, all of these tissues which do have stem cells to regenerate them, led us to think that what goes wrong when you get a cancer or a leukemia or a lymphoma. And Mike Clark, my colleague formerly of Michigan, now at Stanford, and I proposed a hypothesis about 7 years ago that what happens in the development of a cancer is that you start with a normal stem cell that starts to have a couple of genetic changes, and because in a tissue, the only cell that makes more of itself and stays at that stage of differentiation is the stem cell, it's the only cell that can accumulate the genetic and other changes that lead to cancer. So we proposed that cancers would be, like normal tissues, generated by their own cancer stem cells, each one specific for the cancer. And I think it's pretty clear now that that's so. We've shown it in acute leukemia, in chronic leukemia. These are human leukemias. We've shown it in breast cancer. Mike did. We've shown it in brain cancers. We've shown it in head and neck cancers. And we're moving as fast as we can, because once you know that you have each of those cancers that self-renews like the normal cell does, but now out of control, it means that you can go after those cells, those tiny rare cells within a cancer, and ask what went wrong. It doesn't do any good to look at the daughter cells they make that don't self-renew, because their genes that they express have nothing to do with the rare cancer stem cells. We know now, and Mike Clark just published in the New England Journal of Medicine, that you can look at human breast cancer stem cells and look at the genes they express and have high predictive value of the outcome of those patients, irregardless of the therapy they get. And it extends to lung cancer and prostate cancer and some other cancers. We know that it happens in brain cancer, and again, against the Brownback bill, we found human brain cancers as the only cells that would transfer the disease. These are glioblastoma and medulloblastoma from the brain of a human into the brain of the mouse. They only grow in the brain of the mouse. So they require something in that brain environment to keep them growing. Because we can get out those cells and look at the genes they expressed and look at the abnormalities that occur, we think that we can reconstruct it. And as an immunologist, I think we can find new ways to diagnose or treat it. I know this sounds like a lot of hope and hype, but that's where this field is going. That's where stem cell thinking is taking us. So I mentioned a little while ago that there was a class of cells called embryonic stem cells. That's actually a misnomer, and I want to correct it a little bit. This will be important for your article. The embryologists of the 19th century and most of the 20th century didn't call the first stages of development of a fertilized egg as an embryo. They called it an embryo after the fertilized egg. What happens is in the ovary, the egg pops out, a sperm fertilizes it. As it travels down the fallopian tube, it divides 7, 8, 9 times until it forms an entity we call a blastocyst. And I don't even want to explain why it's called that because I don't understand. But on the outside of it, it has about 100 to 150 cells that are for it to implant, to stick to the uterus and start to form the placenta. Inside, there's 20 or so cells that eventually would make the embryo and the fetus and so on. But each of those cells is not yet determined as to what tissue it'll make. It's only after implantation that it starts, and that's when most of the embryologists before this political era said, that's the embryo, and they said this was a pre-embryo. Nevertheless, it's common parlance, and if I tried to say in Congress that it's not an embryo, they'd say you're just trying to fool us by changing the words. So we say what people commonly use. So those cells in the very middle of that thing called a blastocyst can be removed and cultured. It was done first in mice by Gail Martin at UCSF and Martin Evans in Wales in the 1980s. And that proved extremely important for the study of mouse development and the study of genetic diseases that you could at least study in mice. 1998, James Thompson at the University of Wisconsin modified the technique and did it for the first time with human cells. This was a technical advance, a very important technical advance. And now you could have these cell lines growing to very large numbers, starting with this undifferentiated cell. Now the important thing to remember is those cells remain at that early stage of development. And if you remove the factors that make them grow so rapidly, then they start on their own willy-nilly turning into the different cell types of the body. Some hair cells, some teeth cells, hair follicle cells some of us could use, and every tissue type of the body. That's why it's so important to study these kinds of cells, because they have the genetic instructions somehow to make every cell type that we know in the body. Studying development, therefore, with human embryonic stem cells would be straightforward. Now, where do the human embryonic stem cells come from? Mainly from in vitro fertilization clinics. So these are clinics for people who are infertile. And so instead of having babies the usual way, they put the sperm and the egg together in a test tube, let the development occur, and then put the blastocysts in the mother, usually 2 or 3 at most. But they make 8 or 10 every time. So they are frozen down. Scientists up until 2001 who wanted to work on those cell lines started with the in vitro fertilization clinic. The genetics of those cells are people, mainly white, mainly well-to-do, and always infertile. So if we were going to learn something about development from those cells, we'd be learning from a limited source in our society. It may be that there would be all the genes of diseases like sickle cell disease somewhere there, but it's unlikely, or Mediterranean anemia, or the other kinds of genetic disorders that we'd like to understand, we'd like to treat. So it was with some excitement when it was found by several workers, some in Hawaii, some in Japan, Rudy Yanish at the Whitehead Institute at MIT, that in mice you could take a skin cell nucleus, a cell that's determined to be skin, has the genes on to be skin, the genes off to be anything else, and put that nucleus into an egg of another mouse, removing the chromosomes from that mouse first, so the only genetic information comes from the adult stem cell. Now, skin cell. This was amazing because in a low but significant fraction, the nucleus got reprogrammed back to the early time point, and it was the egg RNA components and protein components that did it. We don't know yet how that works. But what it meant is that you could have from a predefined donor mouse an embryonic stem cell line that you made through that reprogramming process. So Rudy did a terrific experiment. He took skin cells from a mouse that had the genetic immunodeficiency that allowed us to transplant human organs into the mouse. And he put the nucleus in, and he got out a cell line, and it made brain cells perfectly well. It made skin cells, it made hair cells. It could not make the immune system. So the genetics of the skin cell donor read true in the cell line. And he had a disease that recapitulated itself. When he and George Daly got the blood-forming stem cells from that line and put it back in the mouse, it retained that disease, genetic immunodeficiency. So then they did another beautiful experiment. They fixed the gene in the stem cell line. Then they put it in, and although it wasn't a perfect experiment, it showed the point that they fixed the disease. Now, that is what you hear about when people talk about therapeutic cloning. A therapeutic attempt to change the genetic deficiency of some person's somatic cells put into an embryonic stem cell line, fix the gene, and then transplant back into them their own cells. If that's all it were, it would be terrific. But in fact, I think it's much more important if you had the genetics of Lou Gehrig's disease, or genetic Parkinson's, or Huntington's disease, or any of a large number of genetic disorders in cell lines that recapitulated the developmental defect or the pathogenesis of this disease. That's why we're also interested in it. In biomedical research universities, what we really want to do is to begin understanding how these diseases work. I should say to you that the genomics revolution that's led to the sequencing of the human genome has discovered for us many of the genes that we knew were inherited to cause these diseases. But the disconnect is between knowing what the gene is and how a combination of genes leads to a particular disease. Lou Gehrig's disease is motor neuron degeneration. I would say even today we don't know if the genetic defect is in the motor neurons or the cells that nourish it or the connections to the muscle or something else we haven't even thought of. So in order to understand disease development, you would need to fix one of the 3 or 4 or 5 genes in a cell line and say, okay, what does that do? And that's why it becomes interesting not only for us in the biomedical research industry, but eventually, and that's a long time coming, for the pharmaceutical industry and the biotech industry, to begin to understand how genetic defects lead to disease. Now, you would have thought that this would be the kind of triumph that everybody would jump on. But in fact, I was actually head of a National Academies of Science, Engineering, and the National Research Council panel, and the Institute of Medicine, on what's called human reproductive cloning and nuclear transfer, what I told you about, to make stem cells. Bruce Alberts, figuring I was the only person who probably hadn't made up his mind at that time, asked me to head the panel. And what we found was that if you look at all the animal experiments where you take the nucleus from a skin cell, let's say, and put it into the egg, and now when you get to that blastocyst stage, actually implant it in the uterus of the same species, that 99.2% of those fetuses died. They didn't die like a regular miscarriage. They didn't die, just a few days to a few weeks after they're put in. They died pretty much, those that didn't die right away, in mid and late pregnancy. And in cows and in mice and other animals, quite often they would kill the mother. So my panel was made up of a group of people, scientists, one medical ethicist, reproductive biologists, and so on, who all had agreed before we met that we'd find out the facts about this field before we made a judgment, and we'd keep our mouth shut until we had argued the facts. So we could say, and said unanimously, that reproductive cloning is not ready for prime time. Even if you had no objection to it, nobody after the Nuremberg trials would submit a person to that kind of medical experimentation where the fetus could die and over half the mothers that take it beyond mid-gestation die. So we asked for a legally enforceable ban on human reproductive cloning, but to come back and look 5, 6, 7 years to see if things had improved in animal studies so that you could contemplate it without giving our own particular judgments about whether we liked or didn't like the outcome of reproductive cloning. But we did say, from the experiments of Janisch and others, that it was clear that it was a different thing to put the nucleus in, to make a cell line from something that couldn't make an organism, except maybe 8 times out of 1,000, and then genetically defective. But you could learn about the disease and treat it. So we also unanimously asked for this kind of stem cell research to go forward, but because it impacted so many aspects of society, we said before it goes forward, there should be a panel not like ours, a panel of people who are experts in law and ethics and the other aspects that we didn't cover, medicine, in order to help advise leaders in Congress and elsewhere and leaders in the medical field whether this would be a good thing to do. We didn't presume that we knew the answer to that. But in the middle of our deliberations, and right after we had a public workshop on the issues, President Bush, August 9th, 2001, in a remarkable statement, I think, said that we should go forward with human embryonic stem cell research using cell lines made before that date but not afterwards, and that we should not do this nuclear transfer at all. And of course, as president, he could have and did have an executive order so that it related to funding, it was not criminalized. Sam Brownback, right after that, I'm blocking, who's the guy in the House who did it? Weldon. Yeah, Dave Weldon, Dr. David Weldon, introduced criminalization bills in the House and the Senate that it would be $1 million fine and 10 years in jail if you did this research. And that passed within a 2 hours debate in the House. It got stuck in the Senate. And the people who stuck it in the Senate were bipartisan, you should know. Orrin Hatch, Harlan Specter, Ted Kennedy, Dianne Feinstein, Tom Harkin, and these people did it after hearings from us, whereas in the House there were no hearings. So that laid the groundwork for the political debate. The political debate next actually came to California. There were a group of people, mainly in Hollywood, mainly parents of diabetic children, who wanted to see whether this research could help them. And I actually have never seen anything like parents of kids with juvenile diabetes. They are the most aggressive, the most straightforward people moving it. unlike relatives of people with these other diseases. Not that the others didn't want it to help, but I just have never seen that kind of activity. Eventually, in California, Robert Klein got involved in this group, this nascent group. He asked the scientists to help write up the legislation, which we did, to assure that if they did get funding from the state, that the reviews of the science would be expert, not the usual peer review that we've become used to at the NIH, where the peers are top to bottom, but just experts in the field. We also included the proviso that nobody who was on the review panel could be from California or have a close connection and receive funds. And then we did suggest that they get at least enough money to do the reviews. So the campaign began, and I won't go through all the campaign except to say you know the outcome. 59% of Californians voted in favor of this, to set aside $3 billion to be spent over 10 to 13 years. And one of the key events, maybe the key event, a few weeks before the election, George Shultz, in your audience, met with and advised Governor Schwarzenegger, who, instead of staying out of the debate, came out in favor of this bill, Proposition 71. And I think this was further evidence that this is not a partisan issue. This was an issue about states' rights and the rights of people in California to enjoy the best that could come from this very long-term research in terms of diagnosis and treatment of diseases. The proposition was immediately held up to question by lawsuits on constitutional grounds, but most of the groups, two of the major groups, were from well-known religious organizations. So the question has come out: a nuclear transfer blastocyst, is this a person? That's the basis of the debate. Is this a person that has the same rights as born people? The president is very clear. He believes it is a person, and that nuclear transfer would therefore be research with the intent to kill a person. And he came out in favor of the Brownback bill in the Rose Garden, and in fact, 3 times in a row took it to the UN, and the 3rd time they passed a bill which has, of course, no enforcement, but to say this research should not go forward. So that's where we are. Once again, when Congress started looking at these issues, the tide has turned dramatically since California vote. Now, the majority in the House and the majority in the Senate are in favor of funding at least expanded embryonic stem cell research, if not this nuclear transfer research. Not enough to overcome a veto. And so last year, the bill called the Castle-Leggett bill was passed in Congress and was vetoed by President Bush. And I should say, later that day, Governor Schwarzenegger loaned $150 million from the state budget to the California Institute of Regenerative Medicine. And it's because of that, and only because of that, that the funding has begun and many things are going forward. So I think that I've said enough. I think I've told you about the science as fairly as I could, that the politics has disturbing issues, at least for me, of conflict between church and state, and that this is exactly the kind of body that should question me and yourselves completely. Thank you.
The preceding program is copyrighted by Stanford University. Please visit us at stanford.edu.